Welcome, everyone. And thank you very much, Avi, for agreeing to give a Build a Cell seminar. You can get started.
Thanks, Kate, and it's really great to see you and all the friends around this table, this virtual table, and I hope we also get to see each other soon together. I wanted to give, I mean, everyone here is experts in the area of synthetic cells and synthetic biology, but I wanted to give the angle that we've been working on for the past couple of years. We're trying to introduce synthetic cells into therapeutic applications. And more groups, also many people here, are actually taking this angle, and it's really fun to see how the field is evolving. But for me, I think the first proof for us as a community that people are looking at us and actually asking, can synthetic cells become therapeutic molecules? Was this email that I received, and I hope that. Donna Mendrick is not angry at me for showing this email, but she contacted me. This was out of the blue. It's just a couple years back, and it was an invitation to speak to the FDA. We're talking here 2022, so it's just the end of COVID, and so everything was still virtual. And when I asked Donna, who was the associate director for regulatory activities, why is she reaching out regarding synthetic cells? I was sure she was interested in drug delivery. She said, no, we're very interested to see how synthetic cells will evolve into the clinic. And her question was even more specific. She said, we see this as an up and coming technology, and we believe that synthetic cells will be able to give an advantage over existing therapeutics that we have in the clinic. And that really resonated with me, and I was very excited. And when I got on the call, I was sure we'd have maybe 10 or 15 people on, but there were more than 180 people on that call, all from around the FDA. And a lot of the questions were about different aspects of synthetic cells and how they can become therapeutic. So I think it's not only us as a community, the regulatory bodies are already looking at us and are hoping that we actually bring new drugs to the clinic. But for me, really entering this field, I came from the area of drug delivery. And when I entered drug delivery, the paper that inspired me was this paper that was published by Harrington and co-workers. These were a group of clinicians that were looking at how drug delivery systems, specifically liposomes, distributed in the bodies of patients, of cancer patients, after being injected intravenously. And you can see in the screen in front of you three patients, one with a tumor at the base of the tongue, another patient with breast cancer and metastasis in the local lymph node near the shoulder. And on the right-hand side of the image, you can notice a cancer patient with a tumor in the lungs and metastasis in the spine, and also in the bone marrow of the hip. And even though these patients have totally different tumors, after injecting intravenously 100 nanometer liposomes, they accumulated at the disease site. And the biological reason for this is that the blood vessels in these tumors is actually more leaky than the blood vessels in healthy tissue. So the nanoparticles or the liposomes circulating in the blood actually extravasate from the circulation through these leaky blood vessels and accumulate at the tumor site. And of course, if you can run imaging as was done here, you can also bring medicines more efficiently using these nanoparticles to the disease site. And that really astounded me that you could actually target a medicine using nanotechnology to a disease site. And based on this phenomena alone, many nanotechnology-based therapeutics actually reach the clinic. The first one is Doxil. It's a 100 nanometer liposome containing doxorubicin inside it. And in Doxil, what they actually showed is that you can improve the treatment of cancer patients, specifically breast and ovarian and age-related Kaposi sarcoma patients, by using these drug delivery systems that reach the target site. But since then, really many other lipid-based and other polymeric-based drug delivery systems have reached the clinic. And the latest one, I think that we all became very much aware of. On the left-hand side of this screen are the COVID vaccines that are also lipid nanoparticles, about 80, 90 nanometers in diameter, injected locally, as many of us experienced, in delivering instead of a small molecule, as in Doxa, which the Doxa has doxorubicin, and the COVID vaccines, of course, have messenger RNA inside, so mRNA. But all these systems have a limitation. And one of the limitations is that you can only carry what you already manufactured. So you have a limited dose that can actually reach that target site. But more than that, many drugs are really hard to load into drug delivery systems. And specifically, if you look throughout the evolution of drug delivery systems, one type of molecule has been really hard to load into drug delivery systems. These are proteins. And people have been trying to tackle this aspect, but not with a lot of success. So small molecules has been addressed very, very well. Nucleic acids also very well. But when you look at proteins, one, you have a limited amount, a small amount that you can actually load in. And two, many times the loading is not efficient because these drug delivery systems actually interact with the protein itself that is being delivered. And that's where I think many of us in the field began thinking, could synthetic cells actually replace drug delivery systems, become a better therapeutic modality? And what we had in mind at the time were, if we have nanoparticles streaming in the blood, could we have synthetic cells also circulating in the blood, where you'd have molecular machines inside that are being developed by all of us that could actually synthesize and churn out proteins that would be therapeutic proteins. And if the system would be a great system, you could also activate it exactly where you want it inside the body. So I think that the image that we and many others had was this synthetic cell. It has an envelope from the outside that can be hidden from the immune system, and then the system would actually be turned on to start synthesizing a therapeutic protein on site wherever it's necessary. And for us, one of the first examples of a synthetic cell that is already capable of synthesizing a protein, this is work that was done by Nitzan Krinsky, who was a former PhD student in our lab. And what she did is she actually imaged the synthetic cell as it was synthesizing superfolder green fluorescent protein inside it. And what we saw that over a period of approximately 45 minutes, we're able to actually synthesize a protein from its building blocks, from the amino acid. Now, for people that are carrying out this experiment at home, I just want to emphasize this is a superfolder GFP. If you use regular GFP, usually it takes a bit longer. So a couple of hours, maybe up to even 12 hours for the protein to fold and to start seeing actually that fluorescence. But the superfolder folds immediately. So we have here a system. It's like a small factory that we could also use. And Gal Chen, who was a former PhD student in our group and now is a postdoc in Harvard University, and I'll show some of her work very soon, actually imaged these synthetic cells. Now it's a static image of synthetic cell, and she showed there in the micron scale. And what we I think as a community have been able to do is really improve this graph on the right-hand side. It's the number or percent of active synthetic cells that we have in a culture. And when we think about therapeutics, you need to make sure that these particles will be homogeneous, not only by size, but that all of them, or at least most of them, should be able to manufacture that protein that we are promising to deliver to the site. And I think as we go on, and also show just some examples from our work, we also show that we're able to improve the ability to actually synthesize it, having higher percent of protein producing particles or synthetic cells that actually are active. And when we look closer at them, their size is micron scale. So approximately the size of cells inside the body. This is work that Omer Adir did in our lab. He's a postdoctoral fellow today in MIT. But what we also showed is that you can not only go into the micron scale, you can also play with the size of these synthetic cells. So you can go down into the nanoscale. In drug delivery, many times going into the nanoscale is actually pretty important. So you have that flexibility when you come to work with synthetic cells, going from the micron scale of 20, 30, 40 micrometers in diameter all the way down to about 170 nanometers in diameter, where we were not able to go smaller than that. Other groups, I think, have been able to go even smaller. But for us, what we saw that actually limited us in size is the ability to pack the DNA into these synthetic cells. It was not the ribosomes. We were able to actually image the ribosomes. You can see one ribosome right here inside a synthetic cell, but just packing that DNA plasmid into the synthetic cell became mathematically very unfavorable when once you go smaller and smaller in size. And these systems, as we know very well, are very versatile. You can actually synthesize many different types of proteins, and we were interested in proteins that are relevant for human health or for other biological applications. So we synthesize proteins that are enable us to track the synthetic cells inside the body, such as luciferase or tyrosinase, which is an enzyme, or in a more sophisticated system, we actually synthesize it, Pseudomonas excitoxin. This is a highly toxic protein that would kill any host. So if you would encode a cell to synthesize Pseudomonas excitoxin, the cell itself would die, but a synthetic cell can actually synthesize this protein. And what you can see in front of you is a culture of triple negative breast cancer cells with synthetic cells that are actually synthesizing Pseudomonas excitoxin, or PE in short. And these are the green dots that you actually see inside the culture. And around each one of these synthetic cells, you can notice the death zone around it, where these proteins are being released from the synthetic cell, and the PE is actually killing the cancer cells. And just for a number perspective, it has been estimated that between 1 to 10 protein copies of PE are enough to kill a single cancer cell. So this is a highly toxic protein that can be used for therapeutics, but you surely don't want it just traveling or trafficking around the body. And just in comparison to the synthetic cells alone, you can see on the left-hand side the culture, this triple negative breast cancer, 41 cell culture on the two left-hand panels, and the panel with the synthetic cells that are actually synthesizing the Pseudomonas excitoxin. So this system is robust when you look in culture and capable of actually synthesizing a therapeutic protein that will be on site. But again, going back to the main dogma that we had in mind, could we also activate these systems from afar? Meaning have a synthetic cell that travels in the body, and then could we actually turn it on where we want it to synthesize the protein at the disease site? And what the first example we took is we caged the DNA plasmids. We added a cage molecule which is sensitive to light, and then we exposed the synthetic cells to light, which released the cage, and by that allowed the protein to be synthesized inside the synthetic cell. In this case, it's a luciferase system. And you can notice on the right-hand side, or the right bar, is that the system is not perfect. These synthetic cells also have some leakage, but you still have several orders of magnitude more protein manufactured or synthesized when you expose the synthetic cells to light. So the cage is removed and the protein begins to synthesize. And this happens in a dish as we have here, but it also works in vivo, in mice. And this is one of the first proof of concepts that we had, that you can inject synthetic cells subcutaneously, expose them to light, and then have the protein actually be synthesized on site because of your light trigger. On the right-hand side, you can see that there is also some leakage or some manufacturing of protein, but again, it's several orders of magnitude smaller than the amount of protein that is synthesized when you expose these synthetic cells or activate them from afar. So we thought to ourselves, if these synthetic cells can actually be activated and they can emit light, can they also communicate with other living systems? And going back to Omer Adir and the work he did, he noticed that the luciferase we were actually using, which was a renilla luciferase, was not emitting enough photons to activate biological processes that we were interested in. So he replaced the plasmid, and he synthesized Gaussian luciferase, which actually emitted more photons inside each of our synthetic cells. And you can actually see what a dish of Gaussian luciferase synthetic cells looks like. You can actually notice the light with your own eyes. So this is a highly active system, and now we felt that we can actually start working with these synthetic cells and have them communicate with living cells. And the first example that Omer actually wanted to test was a synthetic cell that would talk to a natural cell. But this natural cell has to be one that can actually respond to light. And the simplest example that we had were fungi that are light sensitive. So what Omer did is he plated the fungi in an extremely dark room, and he placed the synthetic cells that are emitting light next to next to the fungi. And once we actually went back and looked at the fungi, what we saw is on the left-hand side is that these fungi that were actually exposed to light that was emitted from the synthetic cell actually propagated inside the dish as we expected, because you expose them to light. But more than that, the interesting aspect was that we had a quorum sensing like system. The fungi need a certain amount of light in order to begin activated. So that is activation threshold. And we saw that only when we crossed a specific concentration of the synthetic cells, as you can see in the middle graph, only then did we start seeing activation of the fungi. And I think it taught us a very important lesson, that sometimes you need to cross that activation threshold in order to start seeing an effect that would be a biological effect inside a biological system. But I would want to go back and actually touch on one aspect that I think is important for all of us as synthetic cell lovers, and it's the membrane of the synthetic cell. One of the things that we noticed is that the UV light actually does not cross synthetic cells the wall of the synthetic cell very efficiently. Now, this may be an evolutionary reason, protecting our body from UV light that's in the atmosphere, in the environment. But we noticed very, very clearly that when we expose synthetic cell to light, the DNA inside is not damaged in the same levels as it is when it's outside. Or just to emphasize this, we took DNA, and you can notice here a gel of DNA, and in row number one, lane number one, we have free DNA that was exposed to UV. Row number two, it's already DNA that's inside the membrane of a synthetic cell exposed to UV, and you can notice that the DNA itself is actually protected. And lane number three is no DNA, but in lane number four is, of course, the untreated. So the DNA is protected inside these synthetic cells, and this is important when we come to actually activate synthetic cells using an external light system, but also when light is being emitted from inside the synthetic cell outward, meaning that membrane of the synthetic cell actually attenuates some of the light either going in or going out. And when we come to calculate the amount of light going in or coming out or being emitted, we need to also account for the lipid membrane that is attenuating some of that light. And just from experiments that we ran in the lab, what we saw is that as a rule of thumb, the longer the lipid tail, so HSPC, for example, which is 18 zero carbon and 18 carbon tail, sorry. you have more attenuation of the light in a longer lipid tail compared, for example, to DMPC, which has only a 14 carbon chain. So with the DMPC, you'll have more light that is actually emitted from these synthetic cells. But when you add a double bond and you actually destabilize the lipid bilayer somewhat, you again allow light to cross either in or out of the synthetic cell. And when we use synthetic cells that need a light application, meaning either need to be activated or emitting light, then you want to also select the lipid bilayer to be one that will allow those photons to exit, or at least enough photons to activate the biological system you want to activate. And just to make sure, what we did is we collaborated with physicists inside our institution, and what we actually were able to see is that when you look at the energy levels of a photon and its ability to cross a lipid bilayer, also mathematically, it shows that the wider the bilayer or the longer the lipid chain, you do get significant attenuation of the light that crosses that lipid bilayer. So that's about light crossing. And just going back and giving one point about proteins that actually cross, we did not develop a very, I would say, sophisticated system as some of the groups here are developing. We developed a synthetic cell that is rather soft, has a soft lipid bilayer, and the proteins can actually cross this lipid bilayer when small ruptures occur in the lipid bilayer, just due to mechanical fluctuations or that we assume occur inside the solution. So we do not have any active system for releasing the proteins from these synthetic cells. And I totally acknowledge that this is one of the limitations of the work that we are doing when we look at synthetic cells. But going back to therapeutic applications, we first wanted to ask what would happen inside a tumor. Could we actually synthesize a protein inside a tumor? And if we first showed in a dish that we can actually synthesize the Pseudomonas exotoxin and actually kill the cancer cells, here we actually looked already inside an in vivo tumor. It's a triple negative breast cancer tumor, so same cell line, but now it actually is growing inside a body in the mammary fat pad of mice. And in the first experiment on the left-hand side was to see if these synthetic cells can actually synthesize a protein. In this case, it's luciferase inside the tumor itself. And what we saw is that not only can the proteins actually synthesize luciferase, but also when you add to the mice, you add luciferin. Over time, you actually see reactivation of the luciferase. So the luciferase is also rather stable inside these synthetic cells and can be reactivated over a period of time. And when you now, instead of synthesizing luciferase, which is only an imaging protein, we actually wanted to test the therapeutic ability of these synthetic cells to actually treat the tumor. And what we did is we injected the synthetic cells directly into the tumor. So this is not a targeted system. And they synthesized Pseudomonas exotoxin A. And now we went and we colored the slides of the tumor tissue. And what we saw very, very clearly is that when we administer the synthetic cells that are synthesizing the therapeutic protein into the tumor, we actually see activation of caspase 3. And what this actually means is that the tumor cells are dying at the site of administration. So we have a small factory, I would say, that is synthesizing a therapeutic protein on site. But going back to Gal Chen, she said, treating cancer is extremely important, but in cancer, usually you look at cells that are dying. And that's one of your main goals. And Gal asked herself if she could actually take the opposite biological effect. Could synthetic cells activate the growth of blood vessels? And what Gal did is she encoded her synthetic cells to synthesize a protein which is an analog of VEGF, and it's called FGF. And FGF actually is a protein that is necessary for the formation of blood vessels. And the first experiment Gal ran, she took HUVEC cells. These are cells that are endothelial cells. They can create a blood vessel, but they need a cue. They need a biological cue, and that's exactly what the FGF does. And she showed that when she incubates the HUVEC cells together with synthetic cells that are encoded for FGF synthesis, these cells start actually forming capillaries or small blood vessels based on that activation that Gal encoded within the synthetic cells. So just the beautiful formation of the blood vessels on site. And when we compare this to the control systems, we see it's very clear that the blood vessels are actually formed. But we wanted a better proof, and this is still ex vivo, so outside the body. And here, what Gal did is she took a supported tissue bed, and she embedded inside this tissue bed also synthetic cells, but also other components of tissue. And what she was able to see is that once she starts encoding for the FGF, not only are the blood vessels formed inside this tissue bed, but also collagen is synthesized, meaning you start activating a pathway which activates all the other pathways around it for synthesizing new blood vessels inside the body. But we wanted even a better example. And here what Gal did is she took now the synthetic cells and she embedded them inside a polymer plug. So the polymer plug is an empty polymer that contains now the synthetic cell inside it. We embed this polymer plug inside the body of mice, and you can see the blood vessels that were actually formed inside the polymer plug. So this is an empty polymer with synthetic cells inside it. After being implanted in the mouse, you can actually notice the blood vessels that are recruited due to this FGF that is being synthesized inside the polymer plug and creating blood vessels. But I think an even more convincing image is the histology image. On the left-hand side, again, is the polymer plug with the synthetic cells. And you can see below it actually the blood vessels that are growing into this polymer plug as if it was a natural tissue. So we have here a hybrid system where the synthetic cell and the FGF is recruiting blood vessels from the body into the polymer plug. And if you look at the right-hand side are our controls, and you can notice that these polymer plugs are just empty. They have no blood vessels that are inside them. On the left-hand side, actually, something interesting happens that the empty synthetic cells, or the ones that are not encoding for a protein, they themselves also have a lot of nutrients that also allow some tissue homeostasis into the polymer plug, but nothing like what we see when we actually synthesize the FGF. But then we started getting questions about immunogenicity. And we ran one experiment using the polymer plug at the time. And what we saw very clearly is that these systems are tolerated well by the body. So we saw that when inside this polymer, or when we checked the blood of the mice, we actually did not see any irregular or significantly irregular activation of the immune system. But we did want to dig a bit deeper into this phenomena. And this is, of course, important if we want to go and translate these systems to the clinic. So what we actually first tested is we incubated the synthetic cells together with multiple different cell types. So either neuroblastoma cells or primary neurons, so cells of the central nervous system, GIST cells or gastrointestinal tumor cells. These are GI cells, or HEX, which are kidney cells, macrophages, breast cancer, the 4T1 cells, and also the HUVEC, the endothelial cells. And while different cell types actually respond slightly differently to the synthetic cells, we see across the board at I would say, at the normal concentrations of synthetic cells, meaning 2 or 4 or 6% of the media supplemented by these synthetic cells, we see that natural cells actually respond well to the synthetic cells, meaning that the two can actually coexist and that we can go for hybrid systems. There is some variance between one cell type to another, but in general, we can say that the natural cells respond well to the synthetic cells, and the two can coexist. So this was viability, the first experiment that we ran. And the second experiment we wanted to do is actually now already in vivo. And here we actually compared two administration routes. We compared synthetic cells that are administered intravenously, or synthetic cells that are administered subcutaneously to the same breed of mouse. These are C57 black 6 mice. And what we ran are different panels of different cytokines in the blood. And we actually tested this over a period of 24 hours. So the first bottom line, which I think is the most important because this is a very busy slide, is that after 24 hours, we see that the synthetic cells, all types of synthetic cells that we tested, actually the immune response decays. And I think that is important for us, meaning how long does the response remain? We see across all our experiments about 24 hours or a bit less until you see diminishing or decay of the immune profiles. The second interesting point is that subcutaneous administration usually activates less an immune response than intravenous administration. And this also can be relevant if you want the system to be recognized by the immune system or not, meaning if you're going for a vaccine or for a therapeutic. I think it is important. But when we went in and just to dig into two types of synthetic cells that we actually worked with, one had pure system inside, the pure synthetic or the pure protein producing extract. And the pure system actually, of course, induces a lesser or a more mild immune response to the synthetic cells compared to the bacterial extract or the lysates that we use many times. While both were tolerated well by the mice, the amplitude of the pure system was lower, or the immune response to the pure system was lower than that of the bacterial lysate. And that, of course, makes a lot of sense. Interestingly, though, we also tried to compare pegylated synthetic cells to non-pegylated synthetic cells. And the reason for that is that in drug delivery, most of our drug delivery systems are pegylated. And very interestingly, we did not see a significant difference, at least in our experiments. And with our PEG concentrations, we did not see a significant difference using the pegylated synthetic cells compared to the non-pegylated. This surprised me very much, and is an aspect that we will have to dig in a bit deeper, I think, regarding the pegylated systems. Do we want pegylated synthetic cells or not? Just interestingly is that the PEG that we use actually for these synthetic cells is conjugated to cholesterol and not to the lipid. And we found that it was easier for us to actually manufacture the synthetic cells with a pegylated cholesterol compared to a pegylated lipid in these synthetic cells. So again, the take-home message from this slide is after 24 hours, we see a diminishing of the immune response, and that the lysate-based system, of course, induces a slightly stronger immune activation compared to the pure system. But we also wanted to test another aspect, and we wanted to compare mice of different origin. So we compared the immune response of bulb C mice to the C57 black 6 mice. We injected both exactly the same dose of the synthetic cell. And what we saw is that some of the biomarkers, some of the cytokines differ between the C57 and the bulb C. Again, I think what this emphasizes from our perspective is that when you come to develop a synthetic cell for a therapeutic application, and when you come to test it in vivo, different biological models will actually respond differently. Unfortunately, we were not able to compare male and female mice. Here we tested only on female mice, but I think a larger study may actually study also the effect of male and female mice to synthetic cells, how they would respond to each one of them. And also within your biological control, you want to make sure that the baseline is actually the right baseline for your experiments. Meaning that in this case, for example, you can see that TNF alpha in the C57 mice was significantly lower activated compared to the bulb C. And of course, if TNF alpha is an important marker for you and you want to activate it using a synthetic cell, then maybe the bulb C would be a better choice of a model. And also, if you compare two, it's not that one has a lower immune response. That means it's better. We then went and tested also the biodistribution after intravenous administration. This was done only in C57, black 6. And here we saw a very interesting phenomena, is that the protein distributes differently in our synthetic cell. The protein distributes differently than the lipids. While we find the lipids on the lower panel primarily in the liver after administration, the luciferase, which is the protein that we were actually synthesizing inside these synthetic cells, we found it primarily in the lungs. And what it teaches us is that most likely these synthetic cells pop, I would say, or rupture inside in the body, most likely in one of the first organs that they see, such as the lung, where the protein is actually released, and the synthetic cells themselves continue, or the lipids themselves traffic to the lipid depot inside the body, which is the liver. And what we see is that after 2 hours, approximately, we have most of the accumulation in the liver of the mice. And what it teaches me is actually that our synthetic cells are not trafficking very well, meaning they have some circulation time, but very rapidly they accumulate inside the sinks inside the body, the sink that we have, either the liver, which is the main lipid sink, or the lung, which is also a sink that we have, but that the two actually separate, most likely rather early in this process, allowing the protein to accumulate in the lungs. And by the way, and then it also, you can see it being cleared through the urine. So that's regarding the biodistribution. I'd like to go back to how we synthesize our synthetic cells. And inside the body, we have a lot of cell cycle checkpoints that exist. And here, a study that was carried out by Shani Ackerman and Noah Sharf, two PhD students in our group, and what Shani and Noga actually they came to me and they said, if we want this to be a therapeutic, if we want synthetic cells to become therapeutics, we have to be able to have reproducible synthetic processes, meaning that we'd actually be able to use a machine to synthesize our synthetic cell. That's one. And two, we have to also be able to scale up the process of the synthetic cell. And they actually worked on both aspects. One is the control of that checkpoint, this synthetic cell cycle checkpoint process. And if we're in the synthetic world, then of course artificial intelligence is the right way to check that our synthetic cells are activated, and which percent of the synthetic cells are actually manufactured and which are artifacts that we have inside our solutions. And the second is the scale up, and they use tools that we have in the lab for scaling up the production of the synthetic cell. So the first thing they employed for manufacturing the synthetic cell is a lipid handling robot. which actually allowed very accurate distribution also of the solutions. But also when we went on and we tested this fluid handling robotic system to manufacture the synthetic cells, we saw that we were able to manufacture them very efficiently compared to manual work, which is very dependent on the user. But this did not solve our scale-up system. And here, Shani and Noga took a mechanical system we have in the lab, which we use in order to break up cells, and they tested it for its ability to actually produce large amounts of synthetic cells. So it's almost like a homogenization system, but it's a gentle one because that's why it's called gentle max, and because it doesn't disrupt the natural cells when you break up tissue. And their thought was, could we actually use the same system also for synthesizing synthetic cells? And they saw that very efficiently. They were able to go from small scales that we were manufacturing in the lab, so they went up 30-fold in the manufacturing capability. We didn't go larger than that because I told them it was already wasteful, but these systems, you can manufacture large amounts of synthetic cells and then also test them biologically. And when we tested the synthetic cells, we saw that they were active. But again, we didn't know how many of them were active. So what we started doing is taking thousands and thousands of images of these synthetic cells, and then we used machine learning to understand if these synthetic cells are actually active. And what we looked at are for an active synthetic cell, you want to have a lipid membrane, that's one, and you want to have a protein inside it. And as we know, many times when we synthesize synthetic cells, we actually get sometimes also systems that do not have a membrane or do not have protein that is synthesized inside. How do we make sure what the percentage of only our of active synthetic cells that are actually in the solution? And here, together with Ido Galil, we were able to study these images and then start building using a neural network analysis system to understand when we get a microscopic image of the synthetic cell, how many of these synthetic cells really synthetic cells, or how many of them are artifacts in our head. So we have all these systems together, and I'd like just to end my talk by saying that why use synthetic cells? Because they really allow us a lot of modularity. And when we bring in robotics and into the system, it also allows us scaling up and also reproducing our own studies for therapeutic applications. And one of the nice things I like about synthetic cells is that you have so much modularity that you can actually choose what to do. This is an example of a undergraduate student that came to work with me in the past, and instead of going all the way and synthesizing protein, he wanted to synthesize short hairpin RNA inside a synthetic cell. He incubated then the synthetic cells together with cells, natural cells, and he actually showed very nice knockdown of in this case it's luciferase that's being expressed inside the cell. So we have a lot of modularity and also a lot of therapeutic promise. So when I ask myself, why use synthetic cells for therapeutics, I think looking just forward, it's we can see multiple applications. One is could be, for example, T cell replacement or personalized vaccines, or even edible synthetic cells for food application. And the advantages, and this is going back to that first slide I showed about the FDA, is that synthetic cells do not divide, or at least the therapeutic ones would not be dividing, and they have a limited lifespan. Meaning, if we compare them, for example, to other systems that are therapeutic and administered to the body, we have a very well-defined system. Synthetic cells are extremely well-defined, and we know exactly what they will make. If we compare them, for example, to natural cells, we can have a system that we know exactly what it will make and what it will not make. And that, I think, is extremely important when we come to therapeutic applications. And more than that, we can also activate our system from afar and maybe also in the future turn off, and I think I've seen some studies turn off synthetic cells to make sure they don't over produce, or if the patient will have an immune response, for example, to the synthetic cell or to the protein that is being manufactured, you can actually turn it off so at least that protein will stop from being produced. And that does not occur either if we look at gene therapy or at cell-based therapy. So synthetic cells actually allow us a lot more control. But I think the greatest promise that we have for synthetic cell is that personalized aspect or personalized angle of synthetic cell, meaning that you can manufacture a protein that is tailored for a specific patient and use that protein for treating the patient. So, with no further saying, I just want to thank all of you for listening, and I'd love to also thank our team that worked on this. I mentioned many of the students. You can see in front of you the Y team, and thank you so much, Kate, again for inviting me to speak today to this beautiful audience. Thank you so much.
Thank you. Thank you so much. That was really inspiring. I particularly like your work because it answers the question of why. Like what we're doing is scientifically fascinating, but also you're making it useful. So thank you. There's a lot of
Yeah.
questions in chat. Can you see chat?
Oh, 1 second. So I will. I'll stop sharing so I can see the chat.
Mhm. If you can read the question before answering, people that watch the recording don't see the chat, so that'll help.
Yeah. So thank you for the compliments. I can already see some of them. Um, okay. Great work. Thank you. When you administer synthetic cells intravenously, how do they get metabolized? What's the half-life in the animal bloodstream? So that's a very important question. I think it goes back to what we include inside our synthetic cells and which lipids we actually use to construct the synthetic cells. We use primarily for the in vivo studies POPC and cholesterol, and both lipids are metabolized by the body. I would say more than that. We know today that, for example, cancer cells, if you incubate them with some of these lipids, will actually take them up and use them also as building blocks. But these are lipids that the body recognizes and metabolizes using regular metabolic pathways. Regarding the protein producing juice that's inside, there we did not follow what happened or how it's metabolized. I would assume that most of these building blocks or molecular machines are recognized by the body and either broken down and then utilized again or excreted from the body. But that's a great point, great question. And I think one of the answers to it is that when we compared, for example, the immune response to synthetic cells with a bacterial lysate or versus a pure system, you can see that the body recognizes better the pure system. It's less immunogenic. That's the way I think about it. And if it's less immunogenic, I assume it also metabolizes some of these molecular machines as part of the pathway. I hope that answers the question. Um, could the light-sensitive system work for non-visible wavelengths on the EM electromagnetic spectrum? Maybe some wavelength that can simultaneously pass through tissue without significantly damaging the organism. So I think the dream is to use radio waves to activate a synthetic cell. That would be a dream. We have not been able. We tried, but we were not able. I will tell you something else, and that we have been doing is actually using X-ray in synthetic cells. And I just didn't have time to show that work. Maybe at a later time. What we load into the synthetic cells are 2D materials. So these are sheets of metal-based 2D materials. And upon being irradiated with X-ray, you actually see an amplification of the X-ray due to these 2D materials. So what we're trying to integrate actually into the synthetic cell are non-natural systems that I don't know how they would be metabolized, but we do see that they have better interaction with energy that is emitted. This is a destructive wavelength, so of course X-rays. But the dream, I think, is of course radio or something similar, maybe Wi-Fi. Um, do you think it would be worthy of developing a human gene expressing system to substitute pure? It might be essential if we want therapeutic applications of a synthetic cell. I think that's a great point. And um, we're not our group. We've done some work on the extract just because of the cost of the pure system. So we try to improve, and we published a couple papers. In fact, I was looking at one of the earlier papers by Nitzan Krinsky. She actually worked a lot on the bacterial extract, and that paper has been downloaded, I think today, like more than 30,000 times. So it's a process, but we unfortunately did not investigate further than that to see if we could also create another type of pure system. I think having a human-based pure system would be very helpful for our field. But you need to. It would be a big investment of time. So it's something that someone that is an expert in the field would have to carry. Unfortunately, we are not such experts in that area. So I would love that. Um, is the extract going to be more immunogenic than pure? Yes, the answer is it depends which type of extract. The bacterial extract is more immunogenic than pure. If we maybe use wheat germ, we don't have wheat germ in our lab, but probably wheat germ maybe would be less immunogenic. I think that is totally something that we'd want to look at when we look at synthetic cells. How can you reduce immunogenicity? And thank you for all the compliments. It was Kate, just for this, what a great and supportive community we have.
We're always here to serve. I think Taysok has a question too.
So wonderful. Wow, I love it. So the related question about the pure versus human expression system, I believe that's asking whether we can have, glycosylation system in the, cell-free system. I don't believe anyone successfully done glycosylation using Golgi and some other ER system yet, right?
So Mike Jewett has some of the glycosylation enzymes reconstituted in extract, but I don't think in pure.
Yeah, so I know his work, but this is a more likely small, oligo, saccharide, attaching to the protein, not the entire, original native, glycosylation done yet, right?
I think so. Don't quote me on that, but I think you're
Because
correct.
I'm not following because I studied, cell-free system 30 years ago. I feel like I'm getting old. And then I didn't follow since that. So I guess that might be holy grail in the future, but I guess both of you not the expert in that one. So my real question is actually, again, I feel old. I studied drug delivery in the company 25 years ago. At the time, we are using hyaluronic acid to deliver protein, and that was success. We got FDA approval, and I'm so glad. And then 25 years later, we are now talking about synthetic cell delivering drugs. To Abby, what is your vision another 25 years from now? What would be the field? How are we gonna, completely change? I mean, what would be the future? That's curious about your perspective on that.
Yeah, so Taysak, first thing I will tell you that you never age. So I will that so you say 30 years, it can't be, but you always look very young and energetic. So that's the
Now
first
I'm over 50, and then I'm not now doing this one for reading the things. I'm sorry, I'm 50, I'm getting old.
Congratulations. I will say this. So just regarding post-translational modifications, the first question you raised, of course, that is important. I think for certain proteins that you want to actually synthesize inside synthetic cells. And one of the studies, Omer, I won't elaborate on it too much, but he put in some components, I would say, of post-translational modifications inside one of our synthetic cells. But going to your second question was, where are we going to be as a field in 25 years? So the right person to answer is Kate, but I will take a shot at it, Kate, if you will allow me.
It's
And
your talk.
it's recorded, so in 25 years, maybe someone will be looking and actually listening what we thought. So I think when we look at how much we've advanced in the past couple years, just if we look at artificial intelligence and robotics, right? It's exponential, and they also, I would say, on the other side, when we look at synthetic biology and the cyborg type of organisms, we're seeing more and more of those. So if you ask me, I think we will actually have patients that are implanted with synthetic cells that are personalized to those patients. We'll see astronauts that need a medicine, and instead of taking a medicine to space, they take a synthetic cell, and then they'll print out which DNA they need, and the synthetic cell will actually make the protein that they need inside the body, or the nucleic acid. And I also think that we will be able to see food that is enriched with synthetic cells. I think that's going to happen very, very soon. And the reason it's so important is that when we look at the food we have today, just because there's so many people on Earth, we see some diminishing in some essential nutrients. And a synthetic cell, maybe it's not the right name to use for food applications, but I think for food applications, a synthetic cell can be much healthier than other forms of cells that people are growing for food applications. Because a synthetic cell, you can choose exactly which nutrients you want to put, which fragrances and flavors, and more importantly, where these synthetic cells will actually be absorbed inside the body. So I think as an area, we're still in that first plateau, and the people around here are pioneers. Kate, Shirley, and everyone else here are pioneers. But we're going to see these synthetic cells become very active, and we see it already that the community is growing, and more tools are also being developed. So in 25 years, I assume these systems will have electronics embedded in them. They will be able to communicate with them. They'll probably be inside the body detecting diseases and also treating them simultaneously. And they'll be much safer, I think, than cell-based therapies, because these systems are exactly what you put in. And so I'm looking forward to that future. And the nice thing also as a community, and if Kate, I know we're 1 minute over time. I'll just say this, that when we look at the right-hand side of the screen and we see the nice compliments that people give, that means, I mean, we have a community that wants to help each other, and we're building each other. And what, this type of symposium and other symposium, you feel a very special feeling in this type of community that people want to give tools to one another, not take from one another. So thank you so much.
You know what? Thanks to you, and thanks to Kate, and thanks to everybody in this room. I'm very optimistic. My vision is something like this: 25 years, regarding drug delivery. I scan my body with my cell phone, and then cell phone basically indicate you have some problem, coordinate whatever. And then I basically, in the cell phone or whatever, design the drugs immediately in situ. And I take that drug, and then the drug target exactly the coordinate the cell phone indicate, and then I cure immediately. That's what I want to see. And I'm crazy, I know, but I guess you can do it. Everyone can do it. And then I'm not aging by the time, because thanks to you, and then I'm probably living 200 years by the time. That's what I want to see.
We're
Yes.
believers, so yeah. Kate,
Sure.
it's yours.
Yes, I agree with both of you. This is an amazing community. This is part of why this field is so good. And yeah, we're building the future that we just talked about. All right, thank you so much. Thanks again for a great talk. Thanks for a great discussion. And
Thanks,
have a good
Kate.
rest of your Monday.
Bye, everyone.
Bye.